Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements for you. We hope you were able to join us last month for the second edition of the Dr. GPCR Summit. It was a great week filled with amazing talks from amazing speakers from all around the world, including early career scientists. Find out more about the winners of the best talks given at the summit by the next generation of GPCR scientists by following us on social media today. Also, you can now watch the talks from the summit on our YouTube channel. Please subscribe today. You will get notified whenever we share new videos, and it is also a great way to support our work. Another great way to support us is by subscribing to the Dr. GPCR newsletter. The upcoming newsletter contains the summit survey. We want to hear from you. Tell us what you'd want us to keep, improve, and how to make Dr. GPCR work for you. Stay tuned for the upcoming Dr. GPCR Virtual Cafe events. Visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And today I have the pleasure of talking to Dr. JoAnn Trejo from UCSD. Hi, JoAnn.
Hi, Yamina. How are you?
I'm great. I'm glad that we finally got a time for us to sit down and chat. Welcome to the podcast. I'm excited to be talking to you. As we do with all our guests, please give us an introduction to who you are as a scientist.
Absolutely. Happy to. So first of all, it's really a pleasure to participate in this podcast and to share a little bit of information about myself, as a scientist, as well as our work on GPCRs. So I grew up in Central California, in the Central Valley of California, and I don't come from a family or a long pedigree of scientists. In fact, I'm the only scientist in my family, and I'm often asked, "How did you get interested in science?" And my response typically is, to me, it was really a sort of a natural inclination. As a child, I was very curious and liked to understand how things worked. And especially if there were things that were broken, I was very interested in figuring out how to fix them. So I think that sort of is something that I was naturally born with, was a natural curiosity of trying to understand, how things work. And that sort of is a natural transition into problem solving, which is a big part of science, right? Is that we're trying to understand complex processes, in our particular field relevant to disease, and trying to break apart those different processes and components so we can figure out where the process has gone awry. And that to me is really fascinating, and I'm very excited about those sort of processes. My sort of formal introduction to science and what it was really about was through one of my high school teachers. Her father was an engineer, professor at UC Berkeley. And I was very fortunate to have him take an interest in me because I was interested in science. And I had an opportunity to visit his laboratory at UC Berkeley when I was in high school. And really fascinating, even though it was mechanical engineering and combustion and alternators and how those things worked, it was really exciting to me. And so that I think also had a pretty big impression on me because I got to learn about science, real science as it goes on in a laboratory, and to meet somebody who was an outstanding scientist. So I call him my first scientist mentor, and that was Professor Anthony Oppenheim. He's now deceased. I went to college at UC Davis, University of California, Davis, which is in the Central Valley in California. And it was about 40 miles from where I grew up. I grew up in Stockton in San Joaquin County. And it was like I had landed on a different planet, living on campus and getting accustomed to the college life was very different from my home environment. I am Mexican American and grew up in a very traditional a Mexican-American family. And so there was a bit of cultural shock, I'd have to say, for the first year. When I started my undergraduate studies, I had the interest of studying engineering because it was really the only thing I was exposed to. And I took the high-level calculus classes and started taking engineering classes. And it was fine. It was very competitive, extremely competitive. You know, I did really well as a high school student, but when you go to college, you're competing with people who also did extremely well as high school students, and the competition was pretty fierce. And then I decided to take a biochemistry course, and I absolutely fell in love with the topic. And so I guess during my freshman year, I switched majors and focused on biochemistry and toxicology at UC Davis, and took biology classes as well. And I really loved chemistry, especially organic chemistry, and really got very interested in that. I also had the opportunity as an undergraduate. Now, this was in the 1980s when there weren't a lot of opportunities to do summer research in laboratories. However, through my connection with Professor Oppenheim, he introduced me to a program at the Lawrence Berkeley Laboratory, which is right above UC Berkeley, up in the Berkeley Hills. And they had a summer program, and I applied to the program. I'm sure with his influence, I was admitted into the program and worked there in the summer, every summer since my freshman year through college, in a laboratory that was really looking at breast cancer and chemicals that influence breast cancer progression. And that was with Jack Bartley and Martha Stamfer, who was there at the laboratory. That also meant that I had to live in Berkeley for the summer for these 8 to 10 week internships. And I was also very fortunate that Dr. Oppenheim had a small little cottage where they lived on their property in North Berkeley. And I was invited to live there, which was great because I didn't have to pay rent. So I was able to save some money during those that I earned during the summer internship. But I think more importantly is I got to see how a professor actually lived. And I'm like, this is a pretty good life. You know, he loves what he does. He teaches, he does research. He's a happy person. You know, that's something that I really want to do. And I think that really exposed me to what life is like as an academic. Now, I didn't know very much about the grant writing and all this stuff, but he was constantly working on papers and constantly working on chapters. And I find today, you know, that's a lot of my time is writing papers. You get that one done, and we're working on the next paper. Currently, I'm co-authoring a chapter for Brody's Pharmacology with Laura Bone and Jim Garrison, and that's been kind of fun on GPCRs and molecular pharmacology. So that's sort of, you know, my background as a scientist. As I was, getting ready to graduate, I knew I wanted to go to graduate school, and I had a lot of people talking to me about going to medical school, including Dr. Oppenheim, and strongly encouraging me to take that path. But it wasn't in my heart. It was I didn't want to be around sick people. I mean, medicine is fascinating, understanding the disease process, which is what I do as a scientist, as a biomedical researcher. But I didn't want to treat patients, and so I decided to apply to graduate programs, and I wanted to study drugs and how drugs worked. And I think that was in part because of my passion and interest in biochemistry and inorganic chemistry. And so I applied to different graduate programs and decided to come to graduate school actually at UC San Diego. And at that time, in the late 1980s, early 1990s, it wasn't an umbrella program like there are today. Many graduate programs are these large programs. It was physiology and pharmacology, and I believe there was maybe 6 to 8 students admitted every year. And we had to take the first 2-year courses with the medical students, which was quite fun and quite competitive. And we got introduced to, organ physiology, essentially. The name of our graduate program was physiology and pharmacology. So you really got good training in physiology, whole animal physiology, as well as human, physiology and pharmacology. And I completed my graduate work with JoAnn Trejo. And in her laboratory at that time, we were studying the coagulant protease thrombin and how thrombin signaled to basically to enhance gene transcription. And at that time, there was, a big interest in understanding early immediate genes. So these are like FOS and Jun transcription factors, and how those factors regulated gene expression. And so my dissertation work was focused on delineating pathways that controlled regulation of gene expression by thrombin, essentially. At that time, we didn't know what the thrombin receptor was. Okay. And so thrombin, as we know, is a very important enzyme. It's the key effector protease of the coagulation cascade. It's essential for life. If you knock it out, you don't develop very well. It's detrimental very early in the developmental stages. It's, obviously very important for blood clotting and so on and so forth, and important for, drug therapy, warfarin, and other things that target coagulation factors to reduce thrombin generation. But we didn't know what the receptor was, and it was not clear what kind of receptor it was. It's a protease, and yet this protease can produce very interesting results, in cells. And during the latter part of my graduate studies, Shaun Coughlin identified and discovered the thrombin receptor, which is PAR1, protease-activated receptor 1, through really elegant studies using Xenopus oocytes and SIB selection, doing mRNA expression, and really showed that it was a phenomenal mechanism, unusual, atypical mechanism of activation, where the protease actually cleaves the receptor to unveil a tethered ligand, which is already present in the receptor amino-terminal domain, but it's masked. And the protease essentially cleaves that amino-terminus, exposing the tethered ligand domain, which then interacts somehow with the receptor and induces a conformational change that then allows the receptor to couple the heterotrimeric G proteins at the plasma membrane. So after completing my studies with JoAnn Trejo, I looked at a couple of different labs. So some labs that were focused on gene transcription, so Keith Yamamoto's lab at UCSF. I also looked at Inder Verma's lab at the Salk Institute, and then Shaun Coughlin's lab. And I was a little bit, hesitant about Shaun's lab because he was so young. He had just discovered this receptor, and but I decided that's where I was going to go. And so I joined his lab in 1992 as a postdoctoral fellow at UCSF, and had an amazing time. It really exposed me to, phenomenal science, colleagues who were, just as passionate about science, and colleagues who came from different backgrounds too. So Shaun was trained as a cardiologist, so we had a lot of cardiology fellows in the lab. He was also involved in the physician scientist training program. So we had MD PhDs, and primarily a postdoc-driven laboratory. And the focus of my project in his lab was really trying to understand how signaling by this receptor, which is essentially proteolytically irreversibly activated, how is signaling controlled? And we knew it gave graded responses. You could get a dose response. So clearly signaling was being, shut off. But it has this tethered ligand, which is essentially, present on the receptor all the time, unlike other GPCRs where it can, it's freely diffusible. So that was really a big focus of my work in his laboratory. And that introduced me to cell biology, because the trafficking of the receptor is very important for controlling its signaling responses. And you have, the initial desensitization, which we see with all GPCRs. However, the sorting of that receptor into endosomes and then lysosomes is absolutely essential for terminating signaling. And if you allow the receptor to go back to the plasma membrane, you get this persistent signaling. And that's, detrimental to any normal physiological response. And what we've discovered later in my laboratory is actually in invasive breast cancer, PAR1 is highly overexpressed, and it has these attributes of persistent signaling, and it's signals like that because it fails to sort to lysosomes for degradation. And at UCSF, having that interest in cell biology also allowed me to interact with, some really phenomenal cell biologists, Reg Kelly, Francis Brodsky. We interacted with the young Mark Van Zastro at that time, and he was just starting his lab, and really gave me, really good background and knowledge in cell biology. And, I think when I reflect on that, I realize that science is so multidisciplinary. I was trained really in a cell signaling molecular pharmacology lab, went to Shaun's lab and really got exposed to cell biology and developed expertise in cell biology and in membrane trafficking, lysosomal sorting, which is very phenomenal. And it's something that I've continued in my own laboratory. And I think, as GPCRologists, we have to go where the science takes us. And so, we're constantly interfacing with different fields. And I think that's actually a benefit and a great thing about working with GPCRs.
That's fantastic. And I was just, I think you had a magical trajectory. Just the fact that you were exposed as a teenager to what a scientist does and what's life as an academic look like. It's just, I think it seems that to me that it did really shape your trajectory, that you very early on recognized you saw yourself doing that same type of work.
Yeah.
And interestingly, before you mentioned Professor Oppenheim and the fact that he was an engineer, and you were mentioning that you really like to understand how events happen, how things happen, I was going to say, why biology and why not engineering? And then you said it, and I figured, okay. So here's my question. Let's go back to the beginning. So the biology part came in undergraduate school when you took that biochemistry class. Were you ever interested in biology before that?
Not really. And I think in part because I wasn't really exposed to it, In high school, we took chemistry, we took physics, and and I can't even remember, maybe my biology teacher was really bad. I don't remember anything about biology from high school. I went to a Catholic high school, to St. Mary's High School. And so, it was, we were taught mostly by nuns and priests, but I don't remember biology. My first exposure to biology really was in college.
That's I think at that young age, as a teenager, having a good teacher is what makes an impact, and sometimes determines someone's path forward when it comes to their career choices.
Absolutely. So, given that, I didn't grow up in a household where education was emphasized. We didn't have books, we didn't have, a lot of those things. And I am here today because of the mentors that I have had, the phenomenal mentors that I have had. And I've been very fortunate to cross paths with, people who really saw promise in me and saw some talent in me and really encouraged me to pursue this career in science. So mentors are really, really important, and they're out there. And, I think young people should realize that there's, you can have 10 different mentors, that mentors serve different purposes. And some mentors are more nurturing. Some people mentor by example, but that's a mentor because they really show you what rigorous science can be like.
I love that you touched on a very important point, and I think the word mentor sometimes scares students and people off, saying, "Well, how do I find a mentor, and who's a mentor?" Mm-hmm. And oftentimes, the best way to go about it is really to talk to people and to see who you can look up to. And as you mentioned, someone who teaches by example, or who someone recognizes something in you and decides to help you out.
Right.
But it seems to me that mentorship is very important to you. How do you pay that forward, what you received from others?
Yeah. So there's a big focus on mentorship. Mentorship is absolutely critical for success in any career, and particularly in academic medicine and science. One of the things we certainly recognize in academia and science is that mentorship is not even, and that is that often women and underrepresented scientists don't have access to good, high quality mentoring. So one of the things that I do just personally is to try to ensure that I'm an effective mentor. And mentoring it's not something that you're just born to do. It's a learned skill. I think Bob Lefkowitz is really a phenomenal mentor. And one of the reasons why he's a phenomenal mentor, and he's somebody that I admire and aspire to be like, is that he knows how to motivate people. We talk about our successes. And in any laboratory, there's also, individuals that aren't successful. That's just the nature of having a lab and working with people. One of the reasons why learning to be a good mentor is important is that when you have more than one person in the room, there's two people, there will be conflict. It's just a natural, inherent thing of any relationship. There will be misunderstandings. There will be conflict of different sorts. And there's a good way to handle conflict, and there's a bad way to handle conflict. And you have to learn how to handle conflict well, just to ensure that everyone's sort of, ultimate goals are being met. And, as professors, particularly in academia, our job is to train, is not only to do really good science, to have a well-funded research program that's doing innovative cutting-edge science, but we're also training postdocs and students. And that's a constant element of our programs. And, so you're constantly getting new people that have, that respond differently to mentorship than other people. So you have to be also very flexible. It's not one size fits all mentoring. It's almost tailored and customized mentoring for each person. A big part of developing good mentoring relationships is getting to know people and developing trust in those relationships, and to know that your mentor has your best interest in mind, that they really, understand, all of the different demands on your time, and work with you to try to facilitate your success in the laboratory. And I feel that has gone, that works much better than the old fashioned way of, just demanding that you do this, and this.
You touched on a couple of very important points. I think when you're a mentor, when you're a head of lab, or when you're a manager, which I don't like the word manager, but let's just use it for the sake of example, you have a responsibility towards your people. Mm-hmm. You basically hold their careers in your hands. Right. And it's like being a parent, making sure that all the different kids have whatever they need for their own development, to develop their own confidence, mm-hmm. but also they're different. And I think that's something that may not be a concept that everyone understands, which is why I think it's important to point out that it is important to make sure that all the students or the postdocs get that opportunity
right.
to build their confidence, to do great research, and having that mentor, having that person saying, well, this may not be the best idea, but go ahead and try it. Let's see what happens, is very important. Then again, as you mentioned, trust is also very important.
Mm-hmm.
Because if you don't trust each other, then any comment that is slightly negative, it's going to be taken personally. And it's not, science is not personal when it comes to the data. The data is the data.
Right. Right.
I love that. So let's move a little bit further into, so you mentioned the PAR receptor and all your work during your postdoc on it. When was the moment when it was discovered that it was a GPCR?
It was basically when they cloned it, when they cloned and they looked at the sequence and compared it to known GPCRs at the time, which are the beta receptors. It's a 7-domain receptor. And I think that was a big, that was like an aha moment. It was like, wow. And then the challenge was, well, how does it work? And it turns out that the PAR1 receptor has a sequence in the amino terminus that's called the pyridine-like domain, which is basically pyridine is a small peptide that's released by leach, and it binds to thrombin and inhibits thrombin's activity. So it was very clear that, okay, so thrombin likely interacts with this pyridine-like domain or sequence, and it's a protease, and it's a serine protease, and there were, your classic, cleavage sites and serine protease cleavage sites, and they just basically tested the hypothesis. If we mutate that to an arginine or a different residue, would you block cleavage? And then I think they moved pretty quickly after that. So the receptor was cloned before I actually arrived in the lab. It was 1991, and I arrived in the lab in 1992.
Okay, because my next question would have been, how did knowing that PAR1 is a GPCR affect your way of looking at the receptor, and how did that affect your work?
Yeah. So, I was really interested in primarily in understanding signal regulatory mechanisms. I think one of the things that I also love about this class of receptors is we very early on discovered that, yes, signaling is regulated by desensitization, but beyond that, signal regulation tends to be, we call atypical. It does not require beta arrestins for endocytosis, for example. They're completely dispensable. That was the first paper I published as an assistant professor. So I took my first faculty position at UNC Chapel Hill, and one of the questions I wanted to address is, okay, so we know this receptor is desensitized by GRKs, beta arrestins, are required for this. Isn't it also important for for internalization, like the classic GPCRs? And so that was a time where Bob Lefkowitz had just knocked out both beta arrestin 1 and beta arrestin 2 in mice, and they had derived these mouse embryonic fibroblasts. And I said, those are the perfect, this is long before CRISPR technology. So this is old school technology. We had the gene knockout, but it was, and also before siRNA was used routinely in the lab. And so we had the genetic knockouts. And I said, this would be the perfect system for me to test this, hypothesis. So it took me about half a day to compose an email to him because I had to be perfect. I wanted to make sure I got my point across. First, I had to introduce myself to him. And it turns out in the Lefkowitz pedigree, I'm like the great-great-granddaughter or something like that, because Shaun Coughlin trained with Rusty Williams, who I think was Bob's first graduate student. Yeah. So I composed this email, I send it to him, and he literally responds within 2 minutes asking for these cells, right? And he said, oh, absolutely. why don't you come on over and let's talk about this and so on and like that. And I said, great. When are you available? I'm available tomorrow. Why don't you come tomorrow? And it was just like, oh my God, like my whole world blew up. And so we got all the cells, the control cells and so on and so forth. We put in our receptor, and lo and behold, endocytosis was just fine. And so that led to, us identifying new pathways that GPCRs utilize for endocytosis. It requires the adaptive protein complex 2 and epsin, and also led us to the discovery of a novel pathway for sorting GPCRs from endosomes to lysosomes that bypass this requirement for ubiquitin. And it's just opened up, huge new discoveries. And, always in the back of my mind, we always show it for, other GPCRs that are expressed in a similar context. So the two systems that we focus on in the lab are endothelial cells, because that's where protease-activated receptors have important functions. And then we can translate our work to vascular inflammation, which is really important. And we're looking at them in a natural context, in endogenous receptors. And then in breast cancer, where this receptor is highly overexpressed and contributes to breast cancer progression. So one of the other things, we discovered is that multiple GPCRs in those cells, so purinergic receptor, P2Y1, histamine receptor, the prostaglandins receptors are also regulated similarly. And so my question is, these pathways that we discover, are they really atypical, or are they just another pathway? And, because, clearly the beta receptor is the prototype, but regulation of the beta 2 receptor is not completely applicable to all GPCRs. And there's so many GPCRs that we haven't even looked at yet.
Agreed, and I love that. Anytime someone says, "Well, we went with the beta adrenergic receptor," I say, "Well, that's great and nice." And yes, there is still work to be done on the beta adrenergic. There are things that we don't know, but it is not the only pathway. It is not the only way of doing these things.
Right.
So I ask this from everyone, and I kind of know the answer. I think anyone listening to this podcast, when it will be released, will know the answer to this. Is PAR1 your favorite GPCR?
Absolutely. 100%.
Did you ever envision working on another system?
So we do look at other PARs. So we've done work with PAR2 and also PAR4, PAR3 a little bit. In one project that we're working on now is it's related to biased signaling by PARs. So protease-activated receptors, like all GPCRs likely, display biased agonism. So depending on the activating protease, you get a different generation of a distinct peptide ligand, which, not surprisingly, interacts with the receptor differently and gives you a different flavor of signaling. So we're looking at this in the context of endothelial dysfunction. So thrombin, clearly during vascular injury or inflammation, thrombin's generated. It cleaves PAR1. It promotes a whole host of inflammatory responses that generally result in disruption of the endothelial barrier. So you get tissue edema, vascular leakage, and so on and so forth, similar to histamine. There's another protease in the coagulation system that actually works to reduce thrombin generation. But this protease is called activated protein C. It actually signals to cells via cleavage of PAR1 at a distinct site. And it gives a complete opposite effect compared to thrombin. So it stabilizes barriers, it promotes anti-inflammatory responses and anti-apoptotic responses. And it does this through interaction with other GPCRs. So I think other GPCRs, for example, the sphingosine-1 phosphate receptor and PAR3 expressed in endothelial cells, are influencing the signaling by PAR1 in this context. And it turns out that the signaling is compartmentalized in caveola on endothelial cells. So endothelial cells have a lot of caveoli, and they sequester receptors and signaling molecules to promote kind of more signaling specificity and efficiency. And so that has something to do with it as well as the compartmentalization of these receptors in these microdomains in endothelial cells. But those other receptors are quite interesting. And so it's really looking at PAR1 and how PAR1 signaling is being influenced by these, we call them co-receptors.
I love it. And I love the way you explain these things. I could see, a whole universe happening, in the endothelial cells where PAR1 is the star actor, but then you have all these supporting roles that are being filled out by other GPCRs to kind of have that balance, that yin and yang, inflammation versus reducing inflammation, which I think is just fascinating.
Yeah. That's a project right now that we're very excited about. We have a couple of manuscripts coming out pretty soon on that topic. So.
We look forward to those. We're really excited about that. You mentioned that you're looking also at PAR1 in the context of cancer as well. What are some of the projects that you're working on currently?
Yeah, so again, this is a really interesting and nice example of how PAR1 has unveiled new biology. So we since the work we did with, Bob Lefkowitz, and I believe that paper was published in 2002 or 2004, and Bob is a co-author of my first paper as an assistant professor in JBC. So, we realized that this receptor has different trafficking patterns. And then we discovered in these invasive breast cancers that the receptors overexpress. So then we started working on trying to delineate, well, how is the receptor trafficked in those cells and so on and so forth. And one of the links that we got based on some work on trying to look at pathways that were non-classical lysosomal sorting pathways was this protein called ALEX. So that was the first kind of discovery, is that, receptors sort through these pathways, and we kind of just, define that pathway. And then some other work showed that there's an additional family of arrestins called alpha arrestins. Okay, so these are, they're highly prevalent in yeast. So yeast don't have beta arrestins. They have another type of molecule called alpha arrestins that are predicted to have similar structure based on predictive biology. So they have your classical beta arrestin N domain, C domain, and the C tails are actually quite divergent. But there is no evidence that these proteins regulate GPCRs in yeast. They mostly regulate transporters and other types of proteins. Then they discovered that there's mammalian homologs of these proteins, again sharing similar structural features with beta arrestins. Make a long story short, we kind of honed in on one of these members called ARRDC3. So this is an alpha arrestin, and it is a tumor suppressor. So especially in invasive breast cancer, its expression, it's either there's a chromosomal deletion or it's silenced through epigenetic regulation, acetylation or methylation of the promoter regions. And so when we started looking in our invasive breast cancer cells, we thought, well, maybe there's a defect in ALEX. So this is this alternative lysosomal sorting pathway. ALEX expression was fine. We sequenced that. We sequenced the receptor from these, cell lines to make sure there were no mutations in PAR1, no mutations in ALEX. And, then we started looking at this ARDC3, and it's like, well, it's absent in invasive breast cancer because it's a tumor suppressor. And so tumor suppressors are being, we don't want this. So we developed, inducible ways of re-expressing this protein. And when we re-expressed the wild type protein, we were able to restore normal trafficking. of PAR1 in these cells. And that was really, it was such a clean system because there was a natural knockout, and then we just did the reconstitution or the re-expression, and we're able to restore trafficking of the receptor. So one of the big areas that we're focusing on is how alpha-arrestin basically elicits its tumor suppressor functions. What pathways is it impinging on? So we just published a paper in Journal of Cell Science that shows that it regulates the hippo-YAP pathway. It certainly is interacting there, and we have some other work that's in progress where we're looking at other pathways, and we're essentially also looking at how this molecule can exhibit diverse functions, like what are the critical determinants of regulating its activity and its function in breast cancer. And again, it's, like beta arrestins, I mean, certainly they're regulating GPCRs, but they also regulate other cargo. I mean, beta arrestins can regulate integrins. So this receptor regulates integrins, transporters, other cargos, and likely the activation of the arrestins are different. For GPCRs, it's more of a, we can trigger arrestin activation through different mechanisms. So that's a big area that we're focusing on in the lab right now.
That's great. I have absolutely zero background when it comes to whatever happened. How do you measure degradation, and how do you measure trafficking of receptors? I mean, I have an idea, but I have never had, concrete lab experience. So I'd like to take a moment and talk a little bit about the tools, the experimental tools that you use in the lab to study these events.
Yeah. So good question. I think we were fortunate with PAR1 because PAR1 is activated through proteolytic cleavage. So there's determinants in the N terminus that allowed us and facilitated the development of really good antibodies. Okay, so a big problem with GPCRs is there's terrible antibodies, and so you have to tag them either with an epitope tag or a massive GFP or YFP, which I don't like. I mean, those things are they're beast. And so in Shaun's lab, Shaun had developed some antibodies, and then as an assistant professor at UNC Chapel Hill, I developed some polyclonal antibodies, and then we recently also got hybridomas to those original weedy antibodies against PAR1 and spent quite a bit of money to develop our own hybridomas and so on. So we've got good tools to follow the receptor. One of the things that we're using are to study the receptor in its natural context. So we have got good antibodies, so we can follow the receptor. That's not a problem. We can IP the receptor. We can, isolate the receptor. Is doing proteomic analysis on trying to identify, different pathway components. So we actually had a nice paper in PNAS published in 2020, where we looked at the phosphoproteome of thrombin-activated PAR1 versus the biased agonist APC-activated PAR1. And you can just see these two pathways completely diverge. And that was a nice study for me. It was the first proteomic analysis I had done, because the expectation is, well, if you stimulate cells, everything's going to be phosphorylated. No, you get all these different changes in phosphorylation, not just temporally, but you see increases in phosphorylation and decreases in phosphorylation, and it's different at different times. And that allowed us to identify some interesting modulators of endothelial barrier function. Some of the tools that we're using and developing in the laboratory are trying to use proteomics, phosphoproteomics routinely in our analysis. We're very fortunate being at UCSD and having colleagues, who can do this on a whim. And one of the, certainly an aspect of science, and I believe everyone would say this, is, it's hard to do good science as a single PI. Everything is very collaborative, a lot of team science. And, in the Department of Pharmacology at UCSD, we have a strong group of investigators, and they're all highly collaborative. So we work very closely with David Gonzalez. He's our mass spec experts. And a lot of that work is really initiated by the students and the postdocs, because they're the ones doing the work. And, it's through their relationships that we get a lot of this work done. And then with Jen Zong, who does the FRET biosensors, she's an amazing collaborator, and has, we have to, get these biosensors into endothelial cells and other cell systems, but she's really very collaborative in helping us conduct some of those studies as well. So tools, Brett biosensors are really valuable tools, because now you can target them to different locations in the cell. So we're also interested in endosomal signaling, so we can target certain biosensors to endosomes. You can target them to lysosomes. You can target them to the plasma membrane. So that's a tool set that's also important.
And who would have thought that, quote unquote, non-specific Brett signal could be such a great tool? The term for those biosensors that are at the endosome, the Rab5 and such, the term escapes me, but it was bystander Brett. That's what I was looking for. Yes,
bystander Brett. Yeah.
Which is phenomenal. And so we've talked a little bit about the tools that you do have to study PAR 1. If you had, a magic wand and you could, make appear new tools that would allow you to get more data faster and advance your projects, how would those tools look like? Or what would those tools look like?
Yeah. So one of the things that, we would, in addition, so we've got really good biosensors for signaling. We're interested in endothelial barrier regulation, both how it's strengthened and then how it's disrupted. And it's really localized signaling that is going on. So, thrombin, histamine, they will cause changes in barrier, but it's very transient. It's very dynamic. It becomes disrupted, and then you get recovery. And some of the molecules that we're looking at affect different aspects of that barrier disruption. Some of them will enhance initiation or the magnitude of the barrier disruption, and others are affecting the recovery. Actually, in fact, we have a paper that is in Cell Science Signaling that'll be published August 31st, where we identified heat shock protein 27 as being a mediator for recovery. And we know very little about the mechanisms. It's not something that just happens naturally. It's controlled by signals to get the barrier to recover. And what's been challenging for us is to be able to look in a very localized area of the cell and to see, what's going on with changes in actin cytoskeleton and or adhesion junction molecules and molecules that are important for controlling barrier regulation. So that is, and that's going to require sophisticated imaging and FRET analysis and lots of tool building, most likely, to get at some of those questions.
I love that, and I love also just the way you're explaining it. It seems to me that you'll need new tools, new biosensors to really look at live events that happen over a period of time, and perhaps being able to distinguish between different molecules that control the signal, but also the length of the signal and the types of the changes, the type of changes that happen in the system.
Yeah.
This is really, really interesting. We're going to move on to my next question, which I ask to everyone, and the answer is always yes, but I always ask guests to elaborate a little bit after that yes. Do you think GPCRs are still good drug targets?
Oh, absolutely. Absolutely. In fact, they're probably better drug targets than we thought they would be, 20 years ago. And that's because of the biased properties of the receptors. The fact that you could fine tune or, activate the receptor to prefer certain pathways and not others. And in fact, for PAR1, this activated protein C, which promotes barrier protection, it actually was on the market as a drug to treat sepsis, and its name was Zygris. Eli Lilly had it on the market. And this is really important because we have very few drugs that can strengthen the barrier. So, often what happens in quite severe inflammatory conditions, such as sepsis, where you have systemic inflammation, is the ultimate pathological event that essentially leads to admittance into ICU units and often mortality, is this breakdown of the barrier. So you get this tissue edema, you get vascular leakage and tissue edema, and they treat patients with diuretics to try and reduce the edema in the tissues and so on and so forth, and that will result in organ failure. And so, at least in our opinion, we feel like GPCRs, they're highly druggable. We now know that they can signal biasly. For PAR1, we know that if you activate it in this certain way, that's how APC activates it, you can strengthen the barrier. And so it really is, an important drug target for that area. Now we're interested in identifying the signals that mediate this barrier strengthening, right? Because for certain targets like PAR1, it's also expressed in platelets, and so if you tweak PAR1 in the endothelium, you're also going to hit the platelet receptor. So the hypothesis is, can we identify a key downstream effector that doesn't have such important broad functions, in the human body as a potential target? And that's sort of, our rationale for doing our work and trying to delineate signaling pathways.
I think, yes, one of the challenges is, the answer to my question is yes, but I think one of the biggest challenges is really to make sure that no matter what kind of drug you create, it is very specific at that specific tissue in that context of that disease. And oftentimes the problem is, let's say you want to hit a receptor in the brain, but nowhere else in the body. And that's the problem, is really, as you mentioned in the beginning, when you said that you really like to understand how things work, especially when things are broken, and how to put them back together. And like that component, when it comes to translating it into GPCR research, is really important, to have specific drugs with less side effects, but also that, specifically hit the receptor at the right time, in the right context, at the right place as well.
Right. Yeah.
Let's go back to that first paper that you published. And I love the fact that, Bob responded in 2 seconds to your email. It is still the case. Whenever you email Bob, typically you get a response in about 2 minutes, which I find just phenomenal. And you mentioned that, that first paper and that first work was one of the aha moments that you had as a scientist, when you discovered that beta-arrestin 1 and beta-arrestin 2 had nothing to do with the trafficking of PAR1. What are their career-defining moments or scientific defining moments did you have during your career?
Yeah. So I think that was one of the first ones. And then the second one is when we made the link between this alpha-arrestin protein and ALEX and its impact on trafficking of the receptor. So I think that was around 2005, and a little bit later, we made that discovery. And then I guess the third one would be more recently work done by a phenomenal postdoc in my lab, Neil Grimsey, who's now an assistant professor at University of Georgia, where, again, this evolved from our work that showed that this receptor utilizes this, alternative pathway. It's not your classic, ubiquitin escort-dependent pathway that sorts most GPCRs into the lysosome, such as the beta receptor. It gets ubiquitinated, it engages with these escort components, and then it gets shuttled through the sequential pathway into lysosomes and degradation. But the conundrum was that when we looked at ubiquitination of a receptor, it was robustly ubiquitinated. And it's like, why would Mother Nature evolve this process if it's not utilized for something? So that led us along this path of trying to understand, well, what's the function of PAR1 ubiquitination? And obviously it's a signaling receptor. So we started, screening for different signaling pathways. And what Neil discovered was that the ubiquitination actually acts as a signal to recruit these adapter proteins called TAB. There's TAB1, TAB2, and they directly interact with P38 alpha and can induce P38 activation through an autophosphorylation activation mechanism. And so that was, really exciting. We published that in Cell Reports in 2018, I believe. And it's, and again, we show that the P2Y1 receptor functions similarly. And again, this is all in the context of endothelial cell signaling. Also P2Y1 receptor, prostaglandin receptor as well. They use these proteins to activate TAB and P38 inflammatory signaling. So that, I think, those would be the three defining moments of my career. So the first paper, showing that arrestins weren't important, that was work done by a graduate student, Mei Pang. Then the delineation of an alternate ubiquitin independent lysosomal sorting pathway that requires this protein ALEX and ARDC3 was work done by another postdoc, Mike Dorres, who's an assistant professor at Hofstra University. And then the third would be the work that Dr. Grimsey did in identifying ubiquitin as a trigger for P38 inflammatory signaling. So
fantastic. Talk about following science wherever wherever it needs it goes.
Yeah. I think that to me also is an example of this receptor has revealed so much about GPCR regulation that is counter to our prevailing knowledge. I mean, if you talk to a lot of people, there's like, oh, GPCRs internalized through beta arrestins. They get, they use ubiquitin to sort to lysosomes. So it's, and I also love the fact that, you followed this receptor to discover alternative mechanisms of trafficking that also apply to a whole other set of receptors. Right. Which, to your point that you had mentioned in the beginning, it does raise the question, what is the canonical, quote unquote, canonical receptor trafficking mechanism that GPCRs use? I think every time I talk to someone on the podcast or, even in the context of the virtual cafes, I learn something new. And I think it just opens up a set of other questions that we can continue working on forever and ever. Which leads me to my next question. If you had advice to junior scientists, being PhD students, postdocs, but even in junior faculty, on how to contribute to the field, what would that be? Yeah, I think they're all likely working in laboratories that are focusing on GPCRs. Is to follow your passion, and I think it's difficult sometimes. Because you think everything's been done. And the bottom line is that, look at for PAR1, for example, there's so much more to learn about these receptors. And then I think sometimes young investigators worry about, okay, well, if I open my lab, I'm going to be competing with my postdoc mentor and other people. The bottom line is just the nature of science will take you in different directions. And it just is. It's like people think differently. So the way they design the experiments, the way they think about the science is different. And that diverse thought and diverse systems and way of approaching the same problem is really important, because it allows us to discover things that we may not have discovered before, and it takes you in it. So this is natural sort of evolution, and I think young people do worry about that. It's like, what am I going to do that's going to be different? I think young people, young investigators also, technology is awesome and technology is important, but don't let the technology drive your science. You should not be developing hypotheses and questions that are really focused on using the technology to answer the question. You should really think of a hypothesis and a question and try to identify a niche area. This is one thing also, is to try and identify an area, like you don't want to study the beta receptor and beta arrestin regulation, okay? Because there's 5,000 papers on that. You want to try and find a niche area. There are 800 GPCRs. There's a lot out there to study, and to follow your passion, to be rigorous, to do rigorous research. My philosophy has always been to try to look at systems where we're studying endogenous receptors, and we can translate the work to important physiological responses in vivo. So we do vascular leakage in vivo, or we do tumor metastasis using mouse xenograft models, and so on and so forth. And if you are able to recapitulate those findings, then you can dive in and get at the mechanism. And so that's really important as well.
I love that. And it is really important advice. And we talked about this a little bit earlier. If I can add to this, collaborations are important. Right. You mentioned how many collaborators you have and how many people contribute. And I think we're fortunate to be in a field where, I want to say, most of the time it is easy to reach out to people and say, hey, this is what I'm thinking. Do you want to grab a coffee virtually for now? But can we put our heads together and collaborate?
Right.
And oftentimes it leads to amazing relationships. And that's also key to success.
Yeah. No, right. That's a really important aspect, is that also science is done collaboratively, and learning how to network. Sometimes it's not comfortable for women and underrepresented scientists to approach people, but it's and it depends on how you grew up and your culture and so on and so forth. But there are people out there who are willing to collaborate with you, and collaborations also, it's like the mentoring. They're relationships, and not all collaborations are ideal. But what you want to do is try to develop good collaborations with colleagues that are trustable, and it may take you a few attempts to find those people, but be persistent. And it's an important skill. It's unrealistic to think that everyone you contact is going to want to collaborate. And collaborations also are a two-way street. It's collaboration, and so you have to give, but you also should be receiving. And so it's a two-way relationship.
I love that, and I think you make a great point when it comes to the fact that it's a two-way relationship, and also to the fact that sometimes, I feel like when you're trying to look for a collaborator, when you're trying to look for a mentor, or when even you take it out of the context of science and you're just looking, quote unquote, for a job, it's similar to dating. You want to make sure that you're comfortable with that person, because if you're not, and if you don't have that comfort, you don't see eye to eye, and you cannot build that trust, because ultimately, once you get to know that person, that other scientist, you're going to work together, and the end result will not only be, answering a scientific question and writing a paper and getting grants together, but also forming that trust. You'll get to a point where you'll just, scramble an email to that person, not even, thinking about the format, and they'll know what you meant, and then they'll be like, okay, I got you. And that's an important key, and you just have to be consistent and persistent at finding and surrounding yourself with those people.
Yeah. Yeah. Yeah.
All right. And on this note,
Okay.
speaking of collaborating and having, finding the right mentor, when you have job openings in your lab, where can people find you?
Yeah. So we have a website. The easiest way is if you want to contact me, is just contact me by email. I am very responsible and very responsive to emails. Typically, when I have an opening, I will post things on social media. But if it's a real, if you're interested in GPCRs and have an interest in learning about PARs and training in my lab is very diverse. We have graduate students, they're phenomenal, as well as postdocs and techs, is just email me directly. So even if I don't, sometimes I have post positions, but sometimes I don't, but I still have positions open. And, we are also very successful in getting trainees their own fellowships. My fellows get, American Heart, predoctoral fellowships, NIH F31s or F32s. And UCSD also has a very large number of training grants. So I think we have something like 44 training grants across the institution. And I think 35 of them are in health sciences. That's where the School of Medicine is, Department of Pharmacology is. And so we have a lot of resources for training individuals. And as I mentioned, one of our missions is to train. So we train students and postdocs all the time. So I'm very open to entertaining more postdocs and having more postdocs. I've had several transition out during the pandemic, which is great. In San Diego right now, biotech is booming, and so a lot of people are getting jobs, which is great. But I do need to refill the pipeline.
Yeah. I love that. And if I can add to that, we also do have on Dr. GPCR a career page. There is a form. So it's drgpcr.com/career. Anyone and everyone is welcome to submit a job opening, and we'll be happy to advertise it and put it on our website. I think it's important to have that job board, whether it's for academic positions or industry positions. And especially I think for industry positions, one of the issues that if you're trained on GPCRs and you don't want to be a professor for any XYZ reason, and you want to keep on working on GPCRs, it's very difficult to look at a job ad and say, oh, this is at company X and they might be working on GPCRs. So that's one of the reasons we created the career page, which is one of the most visited pages on our website. So everyone is welcome to.
Okay, I'll post one then, because we actually do have an opening. So I'll go ahead and post it.
Please submit it. Well, thank you so much, JoAnn, for your time. I really enjoyed our discussion.
Yeah, thank you for the opportunity. It was really great. I really enjoyed talking with you.
Thank you. Bye-bye.
Okay.
Thank you for listening to the Dr. GPCR podcast. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Ines Pinheiro, and Alexa Duran. We look forward to seeing you live at the next Dr. GPCR Virtual Cafe. Visit drgpcr.com/virtual-cafe for more information. Also, please subscribe to the Dr. GPCR newsletter today. You can also find us on YouTube. And if you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Also, email us with any questions and suggestions at hello@drgpcr.com. Until next time, stay safe.